Hi everyone, as we have finished our chapter for today, we're going to answer some diagnostic questions and just to check our understanding. So before we look and discuss the question together, have a go and pause the video and try to answer the question by yourself. So let's look at the first question. It says, which statement is not true of hybridization? Well, let's look at our options. The first one says it is relatively inexpensive. Inexpensive means it is not expensive. It is not expensive to perform, right? We want a statement that is not true. So if we look at hybridization, if we remember what hybridization is, let's remember what hybridization is. Well, hybridization is when we have parents with both have desired traits. So we have, for example, parent A, they have a desired trait, and parent B, they have another desired trait. We cross these parents together to get an offspring that is a hybrid of both these parents. So this offspring has both those traits that the parent has, right? So we call this a hybrid. Now, if we remember, and if we look back at our book, the book clearly states that an advantage of hybridization is that we get organisms that can be bred to have certain combinations of traits. So, does it produce offsprings with specific traits? Yes, it does. Does it cross parent organisms with different forms of a trait? I just explained that. I said that, for example, parent A has a specific trait I want, and parent B has another specific trait I want, and they give me this offspring that's a hybrid. Well, that means that they have different forms of a trait. So that is also a true statement. It can take a long time to be successful. Well, if we look at the disadvantages of hybridization, is that it is time consuming. So it takes a long time to be successful. And it is also very expensive. So we can take out A to be the statement that is not true, because hybridization is not inexpensive. Hybridization is very expensive. So the right answer is A. Let's move on to the next question. Next question says, name the process that scientists use to separate DNA fragments according to size. Well, we talked about this when we talked about DNA tools, and we said that this tool separates DNA fragments according to size. Is it selective breeding? Well, now if we remember, selective breeding was in section 1, and that's when we talked about applied genetics. And selective breeding is the process by which desired traits of certain plants and animals are selected and passed on to their future generations. So obviously selective breeding ain't got nothing to do with DNA fragments. Is it cleaving? Cleaving is cutting, right? And who cuts when we're talking about genetics? Restriction enzymes. So that's obviously out of the way. Is it genetic engineering, or is it gel electrophoresis? Well, it's B. We took that. Gel electrophoresis separates DNA fragments according to size. Right, second question says, select the process in which one type of bacterium takes up the DNA from another type of bacterium. Before we look at the options, let's just look at what the question said again. It wants what? It wants the process. Okay, what happens in this process? We have one type of bacterium, so one type of bacteria that takes up. What does take up mean? Mean that it takes into its genetic makeup. So it takes up the DNA from another type of organism. Is that cloning? Cloning is making identical copies of something, right? Sequencing is to find the DNA sequence, right? Manipulating is changing, right? And we talked about manipulating DNA in different ways. The best one that fits this is transformation. And we talk about transformation when we talk about recombinant DNA. And we said that recombinant DNA is the process in which one type of bacteria will take the DNA from another type of bacteria, and that is called recombinant DNA technology. So the answer is C, transformation. Right, which term explains how humans have been able to produce a wide range of domestic cats? What does that mean? The question is basically saying, how have we as humans been able to produce a wide variety of these domestic cats? Is it by test crossing? Test crossing is just to find the genotype of our parent, so that's obviously out. Is it inbreeding? When we say inbreeding, we're keeping a purebred, right? So we're not changing anything. We want to keep it the same, right? We want to maintain it. So that's not variety. Variety is we have different types of things. Yeah, it is using selective breeding. And selective breeding, we said, is divided into two things, into inbreeding and into hybridization. So the more correct answer would be to choose selective breeding and not inbreeding. The right answer is C. Let's move on to this big question. Obviously, such a scary question. A new breed of cattle has been developed by crossing English short-thorn cattle, which provide good beef. So this type of cattle provides good beef, but cannot withstand hot environments. And the Barman cattle in India, they have high heat tolerance, but they produce poor beef. The new breed, so that means we crossed the English short-thorn cattle with the Barman cattle, and we got this new breed of cattle called the Santa Gertrudis, and it produces excellent beef and can live in hot environments. Which term describes this type of cattle? Is it a crossbred? Is it an outbred? Is it a purebred? Or is it a hybrid? Well, when we talk about a purebred, we're not really crossing two different things, are we? It's a pure line, right? We're keeping the same identical traits or desired traits. So that's obviously out of the way. Outbred isn't something that we took, and crossbreed is not the term to describe the organisms that have been or the offspring of organisms that have been crossed. So the right term is to say hybrid. And hybrids are basically organisms or offspring of organisms that have been bred to have certain combinations of traits. So this hybrid has both traits of the parent. So it can make good beef and it can withstand high heat, right? That's why it's a hybrid. It's got both. Oops, that was a mistake. Nobody saw the answer. Harmful recessive traits can be passed through generations of purebreds. Okay, we're talking about purebred animals as a result of what? Well, we took a little relation. We said whenever we're talking about purebreds, we are talking about inbreeding. Why is that? Because what happens is in inbreeding, or the effect of inbreeding, is that purebreds are maintained and the desired traits are constantly passed on. But obviously, because we're just breeding from the same family, to say, or the same breed, then a disadvantage is that harmful recessive traits can be passed on. So harmful recessive traits can be passed through generations of purebred as a result of inbreeding. because when we inbreed, we get to eliminate the unwanted traits and we ensure the desired traits. We maintain purebreds, but a disadvantage is we get these recessive traits because we're constantly breeding from the same breed. So recessive traits can happen more often. Let's look at the next question. Once a tomato grower observes the desired traits in her tomato, she decides to perform a test cross. What is the purpose of doing a test cross? Well, let's remember, what is a test cross? A test cross is when we cross a homozygous recessive, so I'll just label that as 2 small letter H's, when we cross that with a dominant parent to find out the genotype of this parent. So a test cross involves breeding an organism that has an unknown genotype, right? We have an unknown genotype with one that is homozygous recessive for the desired trait. So we cross them and we look at the phenotypes of the offspring to find out the genotype of the parent, because this parent is dominant, but we don't know if they are homozygous dominant or if they are heterozygous. So to find that out, we do a test cross. So what is the purpose of doing a test cross? To determine if the trait is dominant or recessive? We know that the trait is dominant, but we don't know the genotype of this trait. To determine the phenotype of the plants? We're looking at the genotype. To determine if the plants carry beneficial recessive alleles? Also no, because we're looking at a dominant phenotype. To determine if the plants are homozygous dominant or heterozygous dominant? That is correct. It's D. That is the reason we do a test cross. Right, what is the name for the technology that involves inserting the genes of one organism into the DNA of another organism? Well, we talked about this a couple minutes ago. We talked about the name of the technology that involves inserting genes of one organism into the DNA of another organism. Well, if we look at the first page when we talked about DNA technology, we said that genetic engineering is a technology that involves manipulating the DNA of one organism in order to insert exogenous DNA, which is the DNA from another organism. So that means it's genetic cloning. We didn't take anything about bioengineering. And when we talked about cloning, we said that it's identical copies of something. And transgenetics, if we look back, or if you just open your books and you read up the meaning of transgenetics, we talked about transgenic organisms, and we said that they are genetically engineered organisms, right? So genetically engineered organisms by inserting a gene from another organism. So they are genetically engineered. But what is the name of the technology that involves, what is the name of the technology that involves inserting genes of one organism into the other? That technology is called genetics engineering. But transgenic organisms are organisms made using this technology. We don't have anything called transgenetics. We talked about in our class, or in our book, we talked about transgenic organisms. They are made using genetic engineering. Transgenic organisms are genetically engineered by inserting a gene from another organism. So what is the technology? The technology is genetic engineering. What is an example of genetic engineering? Transgenic organisms. So we don't have anything called transgenics. It is genetic engineering. So don't get those two things confused. Transgenic organisms are made by using the tool or the technology genetic engineering. Which type of protein can recognize specific DNA sequences and cleave? We said cleave is cut the DNA. And whenever we talk about cutting or cleaving the DNA, we are talking about our DNA scissors. And when we talk about our DNA scissors, we are talking about what? When we talk about DNA scissors, we are talking about what? We are talking about restriction enzymes, because DNA ligase is our glue, right? DNA ligase will glue up our DNA fragments together. Transcriptase reverses, or RNA transcriptase would reverse the RNA to DNA, and then reverse transcriptase is what reverses our RNA to DNA, and then DNA polymerase is what's going to add up our nucleotides. So what is our DNA scissors? What cleaves, what cuts DNA? That is restriction enzymes. Which process separates DNA fragments according to size and has many applications in genetic engineering and biotechnology? We answered a similar question before when we talked about separating DNA fragments according to size, and we said that that was gel electrophoresis. Next question. A DNA molecule that has had genes from another organism inserted into it. We have DNA molecule. It had genes from another organism inserted into it. What do we call that? Complementary DNA? Exogenous DNA? Exogenous DNA is the DNA from another organism, right? It's our outside DNA. It's our outsider, hence exo. So that's out. Genomic DNA, and that is obviously not it. Recombinant DNA, because as I said, we are combining genes from other organisms. So genes from another organism, combining genes from another organism is recombinant DNA. Let's move on to the next question. All right, why is polymerase chain reaction, or PCR, one of the most powerful tools used by scientists? Why is it one of the most powerful tools used by scientists? Let's look at number 1, or A. It says it can be used to identify errors in DNA sequences and predict the functions of genes. Well, that is obviously a no, because let's just review what polymerase chain reaction is. Polymerase chain reaction, we can make, or it can be used to make millions of copies of a specific region of a DNA fragment, right? And PCR is extremely sensitive, and it can detect a single DNA molecule in a sample, and it can make millions of copies of it. So we're not identifying any errors, and we're not predicting the functions of genes. PCR, polymerase chain reaction. Chain probably means making more of it, right? We're making a chain, so we're making more of something, millions of copies of something. So try to memorize it that way. Let's look at B. B says it can detect a single DNA molecule, in a sample, and it can make millions of copies of it. Well, B is correct. Let's look at C. Let's see if we can find a more accurate answer. It creates large amounts of recombinant DNA in genetically engineered organisms. When we talk about making large amounts of recombinant DNA, we are talking about gene cloning. So don't get those two things mixed up. Let's look at D. D says it creates DNA fragments with sticky ends that can join with other DNA fragments. Well, that is obviously a no, because we said anything that cuts our DNA, anything that cleaves our DNA, anything that gives us sticky or blunt ends is restriction enzymes. So B is the correct answer. Next question. the task of sequencing the entire DNA in human cells has been completed? Yes or no? The answer is a yes, because in 2003, the entire genome, or the sequence of the entire genome of a person, has been sequenced, and that is written in your books if you go to DNA sequencing. Okay? Next question. Which section of human DNA are unique to every individual? Unique to every individual. The regions of DNA that code for proteins. Well, we said most of the regions of DNA that code for proteins are actually similar in most organisms, not just humans. So these aren't unique. The sections that contain genes, every human has sections that contain genes, so that's wrong. The genes that code for fingerprints. Well, the genes that code for fingerprints. Or is it the non-coding sequences? So a lot of people might get very confused here. But in the book, it is clearly stated that the sections of human DNA that are unique to every individual are your non-coding sequences. And if you look back at the videos and you watch those videos again, you'll see us talk about non-coding and coding sequences. And we discussed that the long stretches of non-coding regions of DNA are unique to each individual. And we talk about that in DNA fingerprinting in the human genome in page 81. So when we talk about the genes that code for fingerprints, they are actually the non-coding sequences. They're unique to each individual. So unlike the protein coding regions of DNA that are almost identical among individuals, the long stretches of non-coding regions of DNA are unique to each individual. When these regions are cut by restriction enzymes, as described earlier in this chapter, the set of DNA fragments produced is unique to every individual. Now, DNA fingerprinting involves what? It involves separating these DNA fragments that contain the non-coding sequences in order to observe these distinct banding patterns. So which sections of human DNA are unique to each individual? Those are the non-coding sequences. You might get confused and say the genes that code for fingerprinting, but DNA fingerprinting is actually just taking these non-coding sequences and putting them into gel electrophoresis, or first cutting them and then putting them into gel electrophoresis to observe the distinct banding that happens. So the sections that are unique to each individual are the non-coding sequences. Next question. Which field of study involves the careful storage, organization, and indexing of data? So we're talking about a database, right? On DNA sequencing, is it algorithms? No. Is it bioanalysis? Also no. Is it microarray analysis? Well, that just doesn't make any sense now, does it? Is it bioinformatics? Yes, it is. Why is it bioinformatics? We said that bioinformatics is this storage, cataloging, or maintaining and storing a database and organizing the DNA sequences. So when we talk about the careful storage, organization, and indexing of DNA, or making a database or indexing data on DNA sequencing, we're talking about bioinformatics. Whoops. Next question. If the genome represents the words in the dictionary, then the definition and uses of these words is represented by the haplotype? No, because the haplotype is the combination or the variations of DNA, right? So haplotype is the linked variations in the human genome. Chromosomes, they have our DNA coiled around proteins. Our DNA is basically our DNA is found in our genome, right? So that means it's proteins, right? And we talked about this in the last video when we talked about if genes or the genome represents the word, then proteome, and proteome means all the proteins found in a cell. That's a proteome. They provide the definition of these words and how these words are used in a sentence. So the answer is D, proteome. Moving on, next question. Let's look at the following images. These are a result of what process? Well, if we look closely, we can see that we use homozygous recessive as a parent in both of these. And we see that we have a heterozygous white grapefruit and a homozygous white grapefruit. They have just made a mistake in the image here. So these are all supposed to be capital letters. Oh yeah, they are. Okay, cool. Sorry. We're just gonna act like we haven't seen that answer. Okay? Okay, perfect. So we have the homozygous recessive, and this is our small letters. There we go. And then we have, in one, we took the heterozygous white grapefruit and we took the homozygous white grapefruit and we did a cross. And we found out the phenotype of the offspring, and that helped us to identify the genotype of the parents. And if you go back to your book, this exact image, this exact example is given under a test cross. And in fact, in our lesson, or in the chapters that we are taking now, this is the only cross we will face, which is a test cross. Any other option is incorrect, unless they're talking about previous lessons and the previous chapters we took in the last terms. But whenever you face a cross this term in our curriculum or in the material that we're covering this term, the only cross that we're looking at this term is a test cross. Oh my goodness. Next question. What is the role of the molecule shown in DNA cloning? Right, so is it to carry the foreign DNA into the host cell? Is it to identify the source of DNA as foreign? To identify, and when they said what is the role of the molecule shown in DNA cloning, they basically mean this recombinant DNA. So what is its role in DNA cloning? Is it to identify the source of DNA as foreign? Obviously not. To identify the host cell that has taken up the gene of interest? Also not. To make the foreign DNA susceptible to distinction with enzymes? Also not. What this recombinant DNA does, it helps us carry the foreign DNA into the host cell, because we said that we can't just insert our DNA however we want. We got to put a plasmid with it, right? We've got to put our taxi with it, our vector, our plasmid, and once our vector, which is our plasmid, has connected with our gene of interest, it creates this molecule called recombinant DNA, and this recombinant DNA can then be carried into the host cell. So what does recombinant DNA help us in doing? What does this molecule help us in doing? It helps us to carry foreign DNA into the host cell. So the answer is A. What is the genotypic ratio of the offspring in the cross at the right? Well, we see that we have, when we talk about ratios, you really have to simplify them, okay? You have to put them in the simplest form. So let's look here. We have 4 boxes, right? So it's going to be a ratio out of 4. So here we have 2 grapefruits that are white, because they said heterozygous white grapefruit, so white is a dominant trait. Since we have the dominant allele here, these are going to be 2 white. So we have 2 white for 2 red because these are homozygous recessive, and we said that if they're homozygous recessive, we'll have a red one, so it's 2 to 2, and then we've got to simplify that, so it's going to end up 1 to 1. So the answer is 1 to 1. And if we look at the 1 to 2 to 1, we took this when we talked about incomplete dominance. If you remember last year, when we took the questions about incomplete dominance and complete dominance, we had crosses that would look at 1 to 2 to 1 phenotypes. If we look at C, it says all homozygous recessive. Well, they're not all homozygous recessive because we have 2 that have a dominant trait. All are heterozygous? No, because we have 2 that are homozygous recessive. Next question. A person wishes to raise pigs with black fur, the dominant trait. So black fur is dominant. She selects a male black pig and performs a test cross. Well, if we have a male black pig, that means it has the dominant trait because we said black is the dominant trait. And she wants to perform a test cross with a female that has white fur. If dominant is black, that means white fur is homozygous recessive. So this will be homozygous recessive. This will be homozygous dominant, and we can have 2 genotypes, either a homozygous dominant or heterozygous. Right? So she wants to do a test cross to determine what is the genotype, what is the black pig's genotype if the offspring are white. So she says that she did a cross between this black-furred pig and this white-furred pig, and black is dominant and white is recessive. Now she says, we want to know the genotype of this black pig if any of the offspring are white. So once we did the cross between the parents, we had offspring that were white, right? So the way you're going to answer this question is you're actually going to try to insert homozygous dominant, and you're going to try homozygous recessive, and you're going to try heterozygous. She says that at least one of, so she said if any of the offspring are white. So there are offspring that are white. So you're going to check first with homozygous dominant. If you cross a homozygous dominant with a homozygous recessive, will one of the offspring be white? If yes, then the genotype of the parent is homozygous dominant. If not, you're going to try heterozygous, and if one or more, if any, of the offspring are white, that means that is the right genotype. Let's try it and it'll make more sense for you. So let's try the homozygous dominant. And let's do a cross, and we go this way and then this way. So I'll just fill that in right away very quickly. So if we look here, all offspring have the dominant allele. That means all of them will be black. But in the question they said, what is the genotype if any of the offspring are white? So I want, the genotype is supposed to give me white. So that means this is wrong. Let's try heterozygous. If we cross a homozygous recessive with a heterozygous, it's supposed to be a B, what will our offspring look like? Well, if we look here, 2 of them have the dominant allele, but the other 2 are homozygous recessive. And we said that homozygous recessive is for white fur. So that means the genotype of this black pig is a heterozygous. So that's B. Oh my goodness. Okay, so let's look at the question. How do researchers distinguish between the bacterial cells that contain the recombinant DNA and the ones that do not? Well, if we remember when we talked about recombinant DNA, we said that we add to the recombinant DNA an antibiotic. And the example that we took was AMP, I think it was, epinephrine, I believe. So we put an AMP, which is ampicillin, which is an antibiotic. And we said that we put this recombinant DNA into a petri dish that has antibiotics. And if the antibiotic kills this bacteria, that means it doesn't have the recombinant DNA. Because if it did have the recombinant DNA, it would have the antibiotic. And so therefore, it can live on an antibiotic petri dish. It will not die. So let's look at the answers that has this definition. A says they observed the 2 types of cells under a microscope. That is absolutely wrong. They tagged the recombinant DNA with a fluorescent dye. No, we said that they put an antibiotic into the recombinant DNA. They use an antibiotic to kill the cells that do not contain recombinant DNA. That is correct. They do not use gel electrophoresis. So they use the antibiotic and they kill the cells that do not contain the recombinant DNA, because the cells that do contain the recombinant DNA will survive. Next question. Which is not yet a use for transgenic organisms? Well, this is going to be a bit of a tricky question if you have not focused with the examples of biotechnology. So go ahead and just look back and read about transgenic organisms in our book again. And you'll find that we can make plants that are resistant to insects and viruses. So that is possible. And bacteria, and we can create bacteria that decomposes oils and garbage. We can do that. We also created animals that secrete enzymes that are useful to humans. And you can look back at those examples in page 78 when we talked about transgenic animals, and you can look at, for example, how transgenic livestock have been produced to improve the food supply for the human health. So that they secrete some enzymes that are useful to humans. So, for example, transgenic goats have been engineered to secrete a protein called antithrobin 3, which is used to prevent human blood from clotting during surgery. But the thing that they have not yet used is animals that can produce organs for organ transplants. If you read in the end, they say in the future, transgenic organisms might be used as a source for organs and organ transplants. So the answer is A. It has not yet been a use for them. It might be in the future. We don't know. Which transgenic species could pose a potential threat to other organisms? It should have threat to other organisms. Goats that secrete proteins used to prevent human blood from clotting? No, because this is a positive thing, right? When we talk about a threat, we're talking about a negative thing that doesn't affect the organism itself, but it affects other organisms. So if goats are helping us to stop our blood from clotting, that's a good thing. Cotton that is resistant to infection, that's a good thing. Chickens and turkeys that are resistant to disease, that's a good thing because we're eating them. We don't want them to be sick, right? We don't want them to be carrying diseases. But bacteria that is resistant to antibiotics, imagine you get infected with this bacteria and you can't kill it because it has a resistance to antibiotics. Well, that is a threat, and a threat to a lot of organisms. Imagine you have chickens or turkeys or horses or dogs or humans that have been infected with a certain bacteria and they can't kill it. They can't get rid of this bacteria because this bacteria is resistant to antibiotics. It cannot be killed, right? So that's not a good thing. That is a threat to other organisms, and it could pose a potential threat. Let's talk about why has the Food and Drug Administration halted clinical trials using gene therapy, and we talked about this in our last video, and we said it was because inserting genes is done by viruses that infect the patient's cells. So we said that we use this viral vector, which is a virus, and the problem with this virus is that when you do inject it into humans, it will get the normal gene into the humans, but the problem is that it infects the patient's cells and it caused the patient to die, which is why they put a halt to this clinical trial. It was not because the clinical trials affected the body's response to drugs, and it was not because it was a risk of producing a transgenic human, and it was also not because doctors are able to take advantage of genetic variations. It was because inserting this gene that is in a viral vector, in a virus, this virus is going to affect the patient's cells, and it actually causes the patient's body to have a reaction for this virus, and it causes this patient to die, which is why they put this clinical trial to an end or stop. And that is it for these revision questions.